Hi everyone, welcome to the structural biology course again. We are going through the structural biology techniques where we are discussing crystallography techniques. Today we are going to discuss few very critical things like Ewald sphere, how Ewald sphere is connected with Bragg's law and Laue equation which we discussed in the last class, how the structure factor is important, how the structure factor is related to development of electron density and what is phase problem, and also what is the Fourier transformation. So, let us discuss. We will start with the Laue condition for diffraction connecting to Ewald sphere. What we talked about from the concept of generation of reciprocal lattice which we discussed in last few classes, we learned that each reciprocal lattice vector H is an integer combination of the three reciprocal basis vectors A star, B star and C star. So, H equal to H A star plus K B star plus L C star, where the triplet of integers which are H, K, L, which are Miller indices, characterizes uniquely each reciprocal lattice vector. The reciprocal lattice vectors A star, B star and C star can be derived from the lattice vectors A, B, C from the vector products of the real crystals. So, A star equal to B into C by V, B star equal to C into A by V and C star equal to A into B by V, where V is the volume of the unit cell defined by the vectors A, B, C in the real crystal. Now, it is convenient to introduce the vector S0 which we defined as K in the last class, which is parallel to the direction of the incident beam. Its magnitude is set as the inverse of the wavelength lambda of the incident beam. So, absolute value of S0 equal to 1 by lambda. Similarly, the direction of the diffracted beam is described by the vector S which we defined as K out with its magnitude. So, absolute value of S also equal to 1 by lambda. The Laue condition states that the following equality must be fulfilled: S, which is the diffracted beam, equal to S0 which is the incident beam, and H which we already talked about in the last slide, which is, you could see here, it is an integer combination of the three reciprocal basis vector A star, B star and C star. So, S equal to S0 plus H. The Laue condition requires three vectors S0, S and H must form an triangle with two identical sides, which also we discussed. So, one is S, one is S0 and another one is H. So, this is the triangle. Now, if a sphere is constructed with the center at the origin of S0 and the absolute value of S0 as radius, the diffracting conditions are satisfied only if the end point of a reciprocal lattice vector H is located on the surface of the Ewald sphere. So, up to now we are talking about the Laue conditions, which was S equal to S0 plus H. Now, from that relation we develop a triangle. So, the Laue condition required that the three vectors S0, S and H must form a triangle with the two identical sides which are S and S0 like this, and then you have H. So, now if you have this relation, we have to develop a sphere which is constructed with the center at the origin of S0 and with the value of absolute value of S0 as radius. The diffracting conditions are satisfied only if the end point of a reciprocal lattice vector H is located on the surface of the Ewald sphere. The direction of the diffracted beam is then given by the direction of the vector S. Now, by changing the orientation of the reciprocal lattice relative to the incident beam S0, each vector H which cuts the Ewald sphere gives rise to a diffracted beam. So, only if it cuts the Ewald sphere, that would give rise to a diffracted beam which is parallel to the direction of the corresponding vector S. So, if you see here, you will see that S is 0 and H and how they form the triangle. Now, based on Ewald sphere, it is clear that each reciprocal lattice vector can only give rise to a diffracted beam if the reciprocal lattice takes successively different orientation. So, you look at the picture, you see here the center, it is S is 0 and this is H. Now, where it would cut, then you will get the diffracted beam. For a complete measurement of all the accessible diffraction intensities, the crystal must be displaced so that each reciprocal lattice vector satisfied the Ewald condition, and that is why I was talking about that Ewald is like Ewald sphere is so critical. If you look at in the animation, you will understand it much better. So, if you see here, you will see that the white beam you are looking at, this is the white beam, this is the incident beam, and then you see the yellow beams are generated, they are diffracted beam. Here the origin of the diffracted beam has been placed at the center of the Ewald sphere, but in practice the diffracted beam originate from the diffracting crystal, that is from the origin of the reciprocal lattice, and you will see that only when it is touching, then only you get the diffraction. So, in summary, the Ewald sphere is a geometric construction used in electron, neutron and X-ray crystallography, which demonstrates the relationship between the wave vector of the incident and diffracted X-ray beams S and S0, the diffraction angle for a given reflection and the reciprocal lattice of the crystal. And it was conceived by Paul Peter Ewald, a German physicist and crystallographer. Also, the Ewald sphere is used as a tool to visualize the condition under which the Bragg's law is satisfied, and therefore, a reflection, or you could better say a diffraction spot in the collector will be observable. This occurs when the surface of a sphere centered about the crystal with radius equal to 1 by lambda intersects with a point on the reciprocal lattice. So, now we will come to the structure solution, where we will calculate structure factor amplitudes from the merge intensities. So, we have already talked about the diffraction pattern, where we see that there are spots which are either very high intensity or relatively lower intensity or even lower than that. Those would be influencing the calculation of the structure factor. If you see the structure factor and the diffraction pattern, The structure factor F, H, K, L are the fundamental quantities on which the function of electron density depends. These are very important magnitudes, since the maxima of the electron density function rho x y z show the location of the atoms, that is the internal structure of the crystal. So, if you get the electron density function, you could have get the entire information of the crystal. The structure factor represent the diffracted waves which, when colliding with a photographic plate, which is usually used in old days, or a detector which are now used, leave their mark in the form of well defined spots that form the diffraction pattern. And remember, that is why the last class I was talking about how diffraction pattern are important. Therefore, from an experimental point of view, a crystal structure is defined by as many structure factor as spots contained in the diffraction pattern found in the detector or plate which is used for the data collection. So, if you have a lot of spots, all of them is representing, if they are atomic structure factor, and together they are presenting the structure factor. So, if you talk analytically, each structure factor can be considered as a vector with its amplitude and phase. Remember, amplitude, okay, and phase refer to an arbitrary origin of phases. So, if you see, it is a wave where you get the amplitude of the wave, which is the value which will give you the intensity, okay, and then you get the angle, which is phase. Also, it represents the total wave resulting from the cooperative dispersion, which is diffraction, caused by all the atom of the cell in a given direction of space. So, if there are crystal giving rise to real space or direct space, there would be unit cell and there would be information about the atoms. On the other hand, you will have the reciprocal lattice which will come in the spots, you will have structure factors which we are talking about, you will get amplitude and phase. Now, the graphical representation of diffraction waves as vector is equivalent to considering that the waves can also be represented as complex number. How? If you see here, it is the amplitude, this is the angle, the real and imaginary part of it correspond to the projection of the wave amplitude on the axis of the representation. The phase is the angle that forms the vector with the horizontal axis, which act as the origin to which phases are referred. So, you see here amplitude and here phases. Now, the structure factor FHKL is the resultant of all waves scattered in the direction of the HKL reflection by the n atoms content in the unit cell. So, by unit cell your n atom is how many atom is there in the unit cell, and if you remember, we have talked about how the different symmetry define how many atoms are already there. So, as I was talking about the FHKL, the original structure factor is the combination of all the small contribution from each and every atom. So, FHKL equal to summation F I e to the power I phi, where phi is the phase. The term Fj describing the diffractive contribution of each atom is called the atomic scattering factor. So, instead of atomic structure factor, it called atomic scattering factor, because it comes in a proportional way how a atom would be scattered when the incoming ray is coming. The scattering factor essentially describes the amplitude for the scattering contributed by a particular species of atom. Atomic scattering factor, the structure factor FHKL is the resultant of all waves scattered in the direction of the HKL reflection by the n atoms content in the unit cell, as I talked about. FHKL equal to summation Fi e to the power I phi. Each of those waves shows an amplitude proportional to the atomic scattering factor Fj that measures the X-ray scattering power of each atom. The atomic scattering factor is independent of the position of the atom in the unit cell. It depends only on the type of atom and the direction of the scattering, so that it reaches a maximum in the same direction of the incident X-rays and decreases as a function of the angle of departure. So, if you think about, suppose this is an atom. So, it could be coming from different angle, and depending on that the scattering happen. So, when you see the spots, you have definitely remember, or you go back and see that there are spots which are very bright, there are spots which are very light, that is because of this. So, the atomic scattering factor is independent of the position of the atom in the unit cell. It stays in one position, but then it changes when you rotate them, it changes their position. Like it is there in the unit cell, but how the rays are coming and where it is hitting, that changes. So, it depends only on the type of atom which we are going to discuss and the direction of the scattering, as I told, okay, not on where it sits, it on the what type of atom it is. Let us say it is carbon. How? I am going to discuss, and from where the ray is coming. To understand, as I talked about, the variation of the scattering factor, let us take the example of carbon atom. Okay. So, at the angular value corresponding to sin theta by lambda equal to 0, you will see that means here you will see maximum scattering. The magnitude of the atomic scattering factor is always equal to the total number of electrons. So, here the number is 6 and you see carbon have 1, 2, 3, 4, 5, 6 electrons. You know that 1s2, 2s2, 2p2. So, it is on the number, but then the angle change and see how rapidly it is reduced. It decreases strongly, that is why it is written, it decreases strongly as the angular value increases from 0 to 0.2, it nearly reduces half, and at 0.8 it is almost 0. So, here there is a very interesting fact to discuss about. This is a protein structure, and if you look at it, you will see that it have carbon, it have nitrogen and oxygen. They are the main component, hydrogen is there, but when we are considering the scattering, we are not considering hydrogen as a candidate. So, I talked about before, the protein molecule is composed of hydrogen, carbon, nitrogen and oxygen mostly, and then sulfur in cysteine and methionine, the 2 amino acids, and phosphorus comes in the form of phosphate as a post translational modification. Now, very interestingly, if you compare carbon, nitrogen and oxygen, who are the major scatterer, have 6, 7 and 8 electrons, 1 s2, 2 s2, 2 p2, 1 s2, 2 s2, 2 p3, 1 s2, 2 s2, 2 p4. So, you could easily understand now, as you have seen, that the scattering is mostly dependent on the number of electron present. So, carbon, nitrogen and oxygen all are behaving as a similar scatterer. It is very difficult to differentiate between carbon, nitrogen and oxygen. Sulfur having 16 is better, but not a huge difference it create in terms of their scattering ability. So, what I am trying to say is, if you think in the protein, the major population, you will not get any difference, significant difference in terms of scattering. So, that is a problem, especially when you want to find a reference, and later when we will go to the phase problem, I would show you why that is a big problem and how we solve it. So, these are the major molecules and atoms, and I say all the principal component of protein is having similar number of electrons, hence displaying comparable scattering. Coming to anomalous dispersion effect on the scattering factor. In a first approach, the scattering power of the different atom does not depend on the wavelength of the X-ray radiation. However, there are side effects that make them different. How? If the incident X-ray radiation has a frequency close to the natural oscillation frequency of the electrons of a given atom, there occurs the so called anomalous dispersion, which modifies the atomic dispersion factor Fi. In this situation, the expression of scattering factor is modified with two terms, F prime and F double prime, which represent the real and imaginary components respectively of the anomalous fraction of the atomic scattering factor. So, this is what anomalous scattering is, and it would come F equal to F0 plus F prime plus I F double prime. Suppose there is a crystal which formed by the repetition of the atomic model constituted by the pair of atoms, which we are showing you. This is a crystal which is having pair of atoms red and blue. Of course, any crystalline model can be decomposed into as many simple lattice as atoms. Similarly, here you see the lattice, but now before going into further, I want to talk about another very interesting thing, which is the diffraction patterns, which I discussed in detail in the last class, correlated to number of atoms. This is very interesting. So, here there is a single atom and you get the type of spots. 2 atoms, you see how significantly the spots change from this to that. This is 4 and you already start getting spots which we have seen in our actual protein diffraction pattern. These are couples where you have a lot of molecule in one array and you get a spot. So, this is kind of, you could say, like 1D array, in you get 2 there, it is not 2D, it is still 1D, but here you have the molecules which are into 2 groups and then multiple group. If you see here, you already started getting the diffraction pattern look very similarly what we get in the diffraction pattern of a protein. So, this is diffraction pattern of a protein. So, that is how the diffraction patterns are correlated to the number of atom present in the molecule. So, now we will continue with the crystal which is composed by the repetition of the atomic mode constituted by the red and blue atoms. So, now if we see, we consider Bragg's law, the phase difference between the 2 reflected red beams will be 0 because they are same atom, or 360 degree, which is equal to 2 pi radians. Also, in between the blue beams, this would be the same thing, but as there are separation between the 2 lattices, red and blue lattices, there will be a phase shift between the red and blue waves which you could see here. Therefore, the total diffracted intensity will be less than the arithmetic sum of both red plus blue intensities. What is the effect? Now, the resulting amplitude, that is the diffraction intensity, is controlled by the separation between the 2 lattices, that is by the shape of the motif being repeated. But the resulting diffraction geometry is the same as the one produced of each single lattice, because the diffraction geometry depends only on the lattice geometry which is same. So, the red X-ray beams which are being reflected on the red planes of indices Hk fulfill the Bragg's law, and the same is applied for the blue beams on the blue planes Hk. Now, if Bragg's law is fulfilled, a phase shift between waves reflected on planes of the same color shifted A by H in the direction of the A axis must be 2 pi equal to 360. That is how Bragg's law is maintained. For the same reason, the phase difference due to the plane separation B by K in the direction of F in the B axis must be 2 pi equal to 360. But the beams reflected on the blue planes show a shift respect to the red ones by a quantity which is called del phi, that depends on the separation between the 2 lattices. So, basically if it is same lattice, it is same, if it is another lattice, there is a change, the change is expressed by a difference which is del phi. In fact, this offset can be easily calculated using the following rule of 3 proportion applied to the 3 independent space direction. So, there are 3 differences, del phi A, del phi B and del phi C. 2 pi h x by A, 2 pi k y by B and 2 pi L z by C. Now, if you combine those 3 phase shifts in 3 different axes and generalizing to the 3 dimension, you will get del phi equal to 2 pi h x by a plus k y by b plus L z by c. Now, finally, when you take the fractional coordinates assuming x equal to x by a, y equal to y by b, z equal to z by c and replacing del phi by phi, phi equal to 2 pi h x plus k y plus L z. This is what we consider as the formula 1 for our mathematical analysis for the next level. Now, when we get the mathematical expression of the phase, so this is the mathematical expression of the phase. So, once we get the mathematical expression of the phase in terms of the shape of the crystallographic model, now let us see how to arrive at the analytical expression of the structure factor. We know the expression, but we want to achieve here. Suppose that F1 represent the dispersion of the red atom and F2 for the blue atoms. This is the figure where you could see the red dots, red balls represent by F1 and the blue one represent by F2. So, writing it using vector notation FHKL equal to F1 plus F2. Now, according to the scheme, the module of this vector sum will be FHKL equal to square root of F1 cos phi 1 plus F2 cos phi 2 square plus F1 sin phi 1 plus F2 sin phi 2 square. Its corresponding phase referred to an arbitrary phase origin phi equal to tan inverse F1 sin phi 1 plus F2 sin phi 2 by F1 cos phi 1 plus F2 cos phi 2. Now, generalizing for all atoms and taking into account the general expression for the phase from the formula 1, the module of the structure factor will be FHKL equal to square root of summation j Fj cos 2 pi h x j plus k y j plus L z j square plus summation for j atom Fj sin 2 pi h x j plus k y j plus L z j square. So, this is now the formula 2 and the relative phase would be phi equal to tan inverse summation j f j sin phi j by summation j f j cos phi j. We have used the vector graphic representation to deal with diffraction waves, and this is equivalent to considering that waves can be represented as complex number, because as I told it is the vector graphic representation where we have seen that amplitude and the phase. In this type of representation, the real and imaginary parts correspond to the projection of the wave amplitude on the Cartesian axis, and the phase is the angle that forms the vector with the horizontal axis. You could see here, which act as an origin to which phases are referred. Therefore, with those understanding and taking the formula 2 of structure factor, the complex expression from the structure factor will be FHKL equal to summation j equal to 1 to n for the n atom present, Fj cos 2 pi h x j plus k y j plus L z j plus I j equal to 1 to n Fj sin 2 pi h x j plus k y j plus L z j. So, when we get this expression according to Euler's formula, this expression we could write as FHKL equal to j equal to 1 to n Fj e to the power 2 pi I h x plus k y plus L z. Now, you wonder what is Euler formula. So, this is called formula 3, and Euler formula which we have done the conversion. This is named after Leonhard Euler, who is a mathematician and who developed this mathematical formula in complex analysis that establishes the fundamental relationship between the trigonometric functions and the complex exponential functions, which states that for any real number which is x, e to the power I x equal to cos x plus I sin x. So, now you could understand how we do the transformation. So, f h k l as a complex number can be expressed in terms of its real and imaginary components. So, f h k l equal to a h k l plus I b h k l, where a equal to summation f I cos alpha I equal to f resultant cos alpha resultant, and b equal to summation f I sin alpha I. So, this is alpha or phi, it is the phase angle. It sometimes represent differently. F j are the atomic scattering factor and alpha j are the phase angles of the wave scattered from individual atoms. So, as I told, this is an alternate mathematically equivalent representation for the structure factor. Evaluation of the structure factor. So, if we know that the internal structure of any crystal, that is the type of atoms f j that constitute it, and the positions x, y, z of all atoms n content in unit cell, we can immediately calculate now the structure factors f h k l that define the crystal. To do this, it is enough to apply the formula 3, which actually involves calculating the inverse Fourier transform of the electron density function. We know the formula 3, but we do not know what is Fourier transform and what is electron density function. So, we talked about structure factor, which is f h k l equal to summation j equal to 1 to n f j e to the power 2 pi I h x plus k y plus L z, and electron density function, which is rho x y z equal to 1 by v summation h k l minus infinity to plus infinity f h k l, which is the amplitude, into e minus 2 pi I h x plus k y plus L j minus phi h k l. So, to convert structure factor to electron density function, we have to perform inverse Fourier transform. Now, we know about this, but we do not know about what is inverse Fourier transform. So, let us try to understand what is Fourier transform, and it is not with all of you, but when I teach in the class, I call Fourier transformation and people become afraid. So, just to tell you, we are not going into the mathematical complexity of the Fourier transformation, but because you do not have to do it when you are actually solving a crystal structure, this would be done by the software, but more you understand what is happening, better you could control the process. If there is any mistake or anything happening, you could realize them. So, Fourier transform, as we have seen, we have different waves of X-rays superimposed on one another during the diffraction. Now, it is difficult to isolate the contribution of each diffraction event to determine the lattice structure. So, what to do? To solve this problem, we use mathematical tool which is known as the Fourier transform. A Fourier transform is a representation of some function in terms of a set of sine waves. The set of sine waves of different frequencies has orthogonal properties. In general, any function can be represented in terms of a Fourier series, which is just the sum of enough sine waves of the appropriate frequency, amplitude and phase. That is what you want, right. Now, if we have a function that contains superposition of many different waves with different amplitudes, frequency and phase, such as in our case the diffracted X-ray signal, we pick up on the detector in a experiment. What should we do? Here we can perform an inverse Fourier transform, which we talked about, to determine the individual frequencies of the diffracted X-ray waves as well as their relative strengths. To illustrate the notion of stacking sine waves, we are considering an imaginary one dimensional crystal, so that this is a easy case, and by looking at that we could have developed our idea on the complex crystal. The unit cell contains 3 atoms. It is a simple one dimensional crystal where the unit cell contains 3 atoms, 2 carbon and oxygen. From our X-ray diffraction experiment, we determined that the electron density in the unit cell looks like the following. So, this is the thing we get initially. Our first step is to try to represent this electron function in term of sine waves. The first sine wave has a frequency of 2, meaning that the wave repeats twice over the unit cell. So, you have 3 atoms, 2 carbon and oxygen. So, 2 type of atoms. When it is 2, one would be representing the carbon and one would be representing the oxygen. So, one of the peaks correspond to the oxygen and the other the 2 carbons, as we could see here. Okay. So, this was we are taking a simplified one dimensional crystal, but there are 3 atoms, 2 carbons and 1 oxygen. So, now when we are taking the sine waves, we see the first sine wave frequency of 2, one corresponding to carbon and other corresponding to oxygen. Then we take a second sine wave has a frequency of 3, which means that there are 3 repeats of the wave across the unit cell. It has a different phase, that means we start at a different place on the wave, the amplitude is also different, you could see here. Okay. Lastly, we add the last sine wave with a frequency of 5. Now, you could see that 2 of the peaks of this wave are lined up with the carbon atom. Okay. And now, if we take all the 3 and combine them, we will get the original one. In this way, we have just shown that we can decompose. So, from the main we could decompose our original density function into 3 different sine waves. Since different lattice spacing will diffract X-rays differently, each decomposed sine wave will give us a characteristic of the crystal structure. Therefore, by reconstructing the frequency spectrum from the inverse Fourier transform, we can determine the relative strength of different waves that are contributing to form our signals. Therefore, performing an inverse Fourier transform on the signal will give us valuable information on our crystal structure. So, now probably you are getting idea, like we got the main, we decompose into different, then all the different is give us information, then we do inverse Fourier transform. So, in our example you can see that the sum of the 3 sine wave is a good approximation of the original density function. This indicates that we can describe the unit cell quite well using only 3 sine waves, given the correct choice of frequency, amplitude and phase. But in general we would need to use many different sine waves to build an accurate reconstruction of our original function. As we told, we have taken a simple model. Now, if we look at the inverse Fourier transform of the unit cell, we find that the result consists of a series of peaks, the largest of which are 2, 3 and 5 on the frequency axis. These peaks correspond exactly to the sine wave frequencies that we use to reconstruct the unit cell. The height of the peaks also correspond to the amplitude of the 3 waves. So, you get 2, you get 3 and you get 5. In the case of protein crystallography, the aperture function is regularly periodic due to the repetition of the unit cell in the lattice, electron density distribution within our crystal. The electron density is the inverse Fourier transform of the diffraction pattern expressed rho xyz equal to 1 by V of the unit cell summation f h k l e to the power minus 2 pi I h x plus q y plus L z, where V unit cell is the volume of one unit cell and f h k l is called the structure factor. We have already talked about that for a particular set of Miller indices h, k and L. We can do a summation here instead of integrating, because we know we only have reflection at integer values for h, k and L. For each point in a diffraction, there is a corresponding spatial frequency. So, the distribution of a far field diffraction pattern is Fourier transform of the aperture function. Aperture is a opening, often it is adjustable, that controls the amount of light reaching the lens on a camera or other optical instrument which controls the ray. So, coming to the phase problem, we already talked about the structure factor which calculated with formula 3 from the known atomic structure are presented by vectors, the modules and phases. This is the formula 3. Their numerical values corresponding to the so called absolute scale, since they are calculating with the dispersion factor Fj that depend on the atom numbers. However, the conventional situation is the opposite one. We normally pretend to solve formula 4, where I am coming to determine the structure of the crystal by solving the function of electron density at each point of the unit cell. So, as I talked about the formula 4, this is the formula of electron density. For this purpose, we have to measure experimentally the structure factor using X-ray diffraction, which is possible. However, we must remember that experimentally we could only measure their module, the amplitude, and therefore, we have to face the so called phase problem. I talked some time about the famous phase problem in crystallography. Now, we are in the phase problem where we have problem in measuring the angle. The module of the experimental structure factors are related to the intensities of the diffracted beam, but these are in a relative scale, because they depend on multiple experimental aspects such as the crystal dimension and the brightness of the primary X-ray beam. So, from there we have to understand the problem of phase problem. We know now from the spots in the diffraction pattern we could get the structure factor. From the structure factor it is possible to get the intensity, but we need the phase for that. So, as I told, this is called the famous phase problem in crystallography, and how to solve the phase problem would be the discussion of the next class. Thank you.